focus today on Terminologies 101 with Lucy is Amplified Fragment Length Polymorphism, what we call AFLP. This is a molecular marker or a genetic marker or a DNA marker that we usually use to help us do what we call fingerprinting or identification of individuals in molecular biology. So AFLP is a PCR. PCR is another molecular technique that is used to amplify DNA. So AFLP is a PCR based tool that is used in genetics for DNA fingerprinting, and it will combine now what we call restriction fragment length polymorphism and the PCR technique. So it is highly sensitive in detecting polymorphisms in DNA, and these polymorphisms is what differentiates people. We know that we have differences between our DNA among the population. So AFLP PCR helps us to detect the differences between people. We know that only DNA of twins is actually similar, but for the rest of us, we have various bases and base pairs that are not the same. It was originally described by Voss and Zabu in 1993. So how does it work? This technique is, of course, we've said, is based on the principle of selectively amplifying. This is important because we are going to be using adapters that help us to select the different fragments or the specific fragments that we want to amplify. So we selectively amplify a subset of restriction fragments because we have cut DNA into fragments after the digestion of DNA with restriction endonucleases or restriction enzymes. So this is what happens. You have your extracted genomic DNA from the human being, from an animal. Then you use restriction enzymes to do what we call digestion into various fragments. These are the restriction fragments. Then for selective amplification, we have to add adapters. So adapter ligation, we add various sequences to the ends of our fragments. These are what we call adapters. Once you have ligated the DNA fragments, we are going to now do PCR amplification. So here we use selective primers or primers that are specifically complementary to our ligated DNA fragments. And once the primers bind, then we do amplification so that we can have multiple or millions of copies using PCR. Then after that, we run what we call polyacrylamide gels or do gel electrophoresis, because now this one helps us separate our amplified fragments in terms of their sizes. So you can see here we have what we call a fingerprint pattern. So you have 3 samples from 3 individuals, A, B, and C. So the first one yielded 3 fragments after cutting with restriction enzymes. Then B yielded 4 and C yielded 4. And you can see that these ones are exactly similar. This could be twin individuals.